Hey everyone, welcome back to Cells to Cell. My name is Heem, this is Nick, and today we're actually going to be talking about iPSCs. This is like the third type of stem cell. It stands for Induced Pluripotent Stem Cell. Now, Nick, before we go into how iPSCs came about and everything, can you tell the viewers the difference between omnipotent, pluripotent, multipotent, and all of that?
Omnipotent? You mean totipotent?
Yeah, but
Totipotent.
omnipotent also exists, right?
Omnipotent exists?
I think so.
I thought that was something Catholics used to describe God. Anyways.
Oh, wait, it might be.
Anyway, so totipotent stem cells have the ability to differentiate into any type of cell that you can think of. So usually these are really seen at very early, like beginning stages of the embryo. Pluripotent stem cells are also found in embryonic stem cells, but they are a little bit slightly more specialized, where they can still go through all three lineages, but they can't turn into any single kind of cell. An example would be like an ectoderm stem cell. They can only go into ectoderm lineages. And then we have multipotent stem cells, which we see more in like adults. So we actually have adult stem cells that are multipotent, that are able to go down a couple lineages, but they're pretty specialized to where they could only stay. So an example of this, I would say, would be hematopoietic stem cells. They're multipotent. They can go into the myeloid and the lymphoid, or they can create white blood cells and red blood cells for all kinds of circulation, and they can do granulocytes, they can do neutrophils, all that kind of stuff. And then we have just, what was the last one? Multipotent, and then just, what's the one that can just go down one lineage?
Unipotent,
Unipotent.
not omnipotent.
Not omnipotent. So unipotent is able to just, self-renew, and then it can also be creating, new daughter cells that are more specialized for downline functions.
So when you're born, or when you were conceived, when a sperm and an egg come together, and then 150 cells worth of a mass of cells called the blastocyst.
Mm-hmm.
And basically, this is when your cells at the very, very beginning stage are totipotent. They can turn into any sort of cell. And that makes sense because you're growing as a human. You need eyes, you need lungs, you need fingers, you need bones, you need everything, right? So your cells from this little ball need to differentiate into all the different things of who you are, so they're totipotent. And then after a bit, they become pluripotent when they become more confirmed to their lineage. So when you stop growing and you need your bones to stay bones, then they slowly transition from that toti to that pluri to the multi and then to the unipotent. And when you have skin cells, for example, they are unipotent, meaning they're only going to be that skin cell. They're not going to be anything else. But the ability for stem cells to be so amazing is their potency. So when they can turn into other cells. So this one scientist in Japan named Yamanaka, he found out that embryonic stem cells, so the baby stem cells, they're pluripotent and they can turn into a lot of different types of cells, which is really good for medication purposes. So taking those proteins that exist in an embryo and putting them on a normal skin cell turned that skin cell back into an embryo stage. So that's kind of weird.
Yeah.
Like, can you go over that a little bit more and how like a skin cell can turn into?
Yeah, the proteins that he added like in the petri dish and all that kind of different stuff. So he was just adding all these different factors. And these factors were actually driving the skin cell backwards. So usually when you think of stem cells and their stemness, they're going to be self-renewing, they're going to continue forward and creating, new stem cells as they divide, because, if you divided into two non-stem cells, you'd run out of stem cells. So stem cells constantly are regenerating themselves along with more specialized cells. But when you take those, a multipotent stem cell or a unipotent stem cell, such as a skin stem cell, or even just a skin cell, a somatic cell, and you add these, you can drive it back. So it actually like reopens all of the transcription pathways and all of the variability that, you know, it had as it was once an embryonic stem cell. So, you know, rather than just being able to make new skin cells, it might be able to make a neuron or a new muscle cell or, you know, a new hepatocyte or liver cell.
So we've actually used this analogy before, but it's just like if you wanted to be a doctor, you would just put on a white coat, some scrubs, and then you'd be a doctor. If you wanted to be a lawyer, you put it on a suit and you're a lawyer, you know? And the same kind of thing works in this case. So these embryonic stem cells exhibit certain proteins on their outside. And if you take these same proteins and put them on a regular skin cell, that skin cell now thinks it's behaving like an embryonic stem cell, and it's actually going to be more potent. And the 4 Yamanaka factors, which are known to be highly expressed on, so these are protein transcription factors that are highly expressed on embryonic stem cells, are OCT3/4, SOX2, KLF4, and c-Myc. And all these 4 proteins together are so abundant on an embryonic stem cell that this researcher, Yamanaka, thought that if I can take all these same proteins and put them on a regular somatic cell, an adult cell, what would happen? And what happened is these cells started behaving, having characteristics as an
embryo.
Yeah. And it started to differentiate really well.
And what's important to understand is, like, you know, it's one thing to figure out how to drive these cells backwards, right? And that's important because it gives you like a very nice starting point. But what's important to understand is that a lot of scientists are still trying to figure out, once we drive that cell back into an embryonic state, how do we drive it in a new direction that we desire?
Yeah.
Right? So say, you know, somebody who is having, you know, cirrhosis of the liver. Their liver is just, you know, rotting away basically. And you're like, why can't we take, you know, these stem cells, these embryonic stem cells, these induced pluripotent stem cells, you know, and drive them towards becoming new hepatocytes and inject them? Granted, it would take a very long time to go through the FDA to do all this cell therapy, but in general, like, the question is how do you go from that embryonic state into that hepatocyte, into that liver cell? And, you know, it actually comes down to developmental biology and understanding how the embryo and all of those cells are able to differentiate, and how they communicate with one another, and where do all of our very specialized cells come from, and how can we, you know, almost manipulate these cells. And I think it's important to understand when we talk about stem cells on this podcast that, you know, people have a really bad connotation with stem cells. They hear stem cells and then all of a sudden they think embryonic stem cells.
Yeah.
You're killing, you know, you're taking out, you know, cells from a fetus, you're taking the life. That's not how it works. That's really not how it works. We talked about mesenchymal stem cells or mesenchymal stem cells. We've talked about hematopoietic stem cells. We're talking about induced pluripotent cells. And we have embryonic stem cells, but they're from cell lines. It's really, if we didn't have them, they'd be biomedical waste. Like, the cells would just be thrown away. So we use them for science. And we also have umbilical cord blood stem cells that are also very, very useful. So understanding, one, where all of these cells come from, and two, how we can utilize these cells is something that's really important. That, you know, I feel like sometimes people get it kind of misconstrued on like what scientists are actually doing. They think like sometimes like we're like witch doctors, and we're just like casting all this magic and, you know, going against society. But there's a lot of ethical codes, there's a lot of morals that go into the research that is going on both here at Case and all around the world regarding stem cells. And this induced pluripotent stem cell science and driving these skin cells back is so important and such a very great step in science. And it's been 10 years ago, which feels like forever, but it's still such a new science and there's so much potential for it.
When you think about the donor market and how it's so difficult to find donors for a lot of treatments.
Mm.
This kind of solves that problem, dude.
Yeah.
You don't need anybody to donate for anything.
No.
If you have a problem, let's say if you're even balding, you could take your own skin that already falls off all the time anyways, right? Or you can just take a good chunk of your own skin in the lab, turn them into these stem cells, and then transition them from them being a skin cell to a hair cell, and then implant those hair cells. So a majority of the cost of a lot of these stem cell transplants in the future is going to go down because you don't need this whole donor system.
Right.
You can donate to yourself.
It's all going to be allogenic. It's all going to be self.
Autologous.
Or is it autologous?
Yeah,
Is it
allogenic,
autologous?
yeah.
Is it allogenic?
Allogenic
Yep,
is
I
other.
will.
Yeah.
Anyways, it would all be autologous. So, and what would be convenient about like growing those like hair follicles or something, and the way that I think about it is like your hair follicles are already kind of a specialized skin cell. So you could just drive it back like a little bit.
Yeah.
Like you don't need to drive it back all the way, and then you can just reprogram it in the lab to start growing hair and using proteins and in a different way. But
I actually thought about something, okay? So let's say you are a 35-year-old adult. Your skin cells have been skin for the past 35 years, right?
Yeah.
So they are very specialized to be just the skin. Now the genes and all the things that are activated inside of them, they are very good at just being a skin cell. So let's say you take the skin cell, you turn them into an iPSC. So now that it's a stem cell, you can turn it into hair, right? The genes that are active inside of that skin cell for those past 30 years, even though they might turn into another cell, it's going to take a lot for the inside machinery to fix itself to now be a hair cell, you know?
Mhm.
So that's, I feel like, an issue with iPSCs because we had a professor come and talk to us about iPSCs. She told us that iPSCs have a difficult time staying in their lineage. So there's the story, okay? Somebody took a regular skin cell, turned it into an HSC, so a hematopoietic stem cell, and was trying to do research with that. And although it was successful in that turning a skin cell into a stem cell and a stem cell into an HSC, what was difficult for the researchers to do was to keep that HSC as an HSC. It's because the inside machinery of that cell was so used to being a skin cell, over time, that HSC turned back into a skin cell. So even though it's very possible to take a skin cell, turn it into a stem cell, and turn that into another cell, even though that's possible, keeping that cell the same way is very difficult because the machinery inside of it is so used to it being something else for all those years.
how cool would it be though, is if you utilize iPSC science, drove a skin cell back, okay, and then you use like CRISPR or like a lentiviral vector, and you introduced like a knockout gene almost.
Ooh.
And you took away those genes, like what makes it a skin cell. And it would take a lot of work to understand all of the different mechanisms and stuff, but like what's like the 3 biggest genes and how can we knock them out or silence them a little bit to where we turn it into an HSPC because
Can induce the help or something.
right, HSPCs are nothing like skin cells except the fact that they have a phospholipid bilayer and they have a nucleus. Like that's the only 2 things, and the DNA is the same, just so much stuff is turned on and off.
Yeah.
So if you can figure out how to turn things on and off, which is what they do, right? If you're driving a cell from an embryonic state to a more specialized state, you are turning the right things off and you're turning the right things on, or you're keeping the right things on if
Yeah, you
everything's
have to
being
keep them
expressed. So it's figuring out how do we increase the longevity of these induced pluripotent cells, and what would be the applications at that point. And I think that again the sky's the limit, especially when we're talking about all these kinds of stem cells, whether it's mesenchymal stem cells not only being used to help with inflammation, but also find a way to use their abilities to fight bacterial infections, maybe for superbugs or something. Or, when antibiotics don't work, maybe you put in stem cells that, lower inflammation or raise inflammation and cause a higher immune response.
Another thing.
Oh yeah.
Now imagine this,
You got a
okay?
thought?
So, if you're taking, let's say, a shoulder cell, so one of your muscle cells here, okay? You take him out, but you don't want to turn it into another cell. You just want to make it healthier and younger. So let's say you put these Yamanaka factors on this muscle cell, turn it back into an embryo, but then still put it back where it came from. So the inside machinery is still good, but now it's behaving like a younger version of itself.
That would be really cool.
Now, isn't that interesting? Because you're not really like changing anything. You're taking it out, putting in these factors, and putting it right where it came from. So nothing is being confused. The machinery is all good. There's no HLE mismatch, so there's no graft rejection. It's a lot of things that might work out when you think about it.
Yeah, and again, it's only like the Nobel Prize was given out in 2012. I think the science was created like 2006, 2008. So we're almost hitting like 15 years where the world has known about induced pluripotent cells, but it's just in its infancy. I have a feeling in the next 30, 40 years, you're going to be more worried about what cell therapies you're going to get rather than what, small molecule drugs you're going to get or what antibiotics you're going to get when you go to the hospital or when you go to the doctor's office to get a prescription. I really do. I think that stem cells really are the future, and it's a great field to be in. And if you're interested now, it's almost like investing in solar. Like one day we're all gonna have to go to solar energy. Like if you're investing in solar at $3 like a stock, it's gonna be like the next Tesla, hopefully. So hopefully as science, this is the next Tesla. This is the next, up and coming thing that just blows everybody out of the water.
Oh yeah.
And I think it's fun to be in the center and Center of Regenerative Medicine and the National Center and learning about it, and then also trying to share it with you guys. And yeah, I think that induced pluripotent stem cells are going to be, or iPSCs are going to be great for the future, along with MSCs and HSCs.
And it turns out that sometimes using stuff for research requires actual cells. And over the summer, I had the privilege of attending this presentation where somebody was doing research on neuronal stem cells. So what they did to do all their experiments on neurons wasn't actually source neurons from somebody. They used an entire iPSC model.
Wow.
So they actually took an entire vat of stem cells or skin cells, turned them into iPSCs by themselves, then turned those iPSCs into neurons and muscles, and then they stimulated those neurons to stimulate the muscle. And it was awesome. And it was all done through iPSCs.
That's crazy.
So iPSCs don't even need to be used for medication purposes. They can be used just to simulate things.
That's like, we had a guy Cleveland cord blood come in. So they're an umbilical cord bank here in Cleveland. That it's a public donation facility, and they do great things. Dr. He was one of the executives. He was like the head of science. It's like Hoff, Dr. something.
Van Hoff?
Yes. He came in and he was talking about all the different things. Oh, wait, no. I'm thinking about the wrong guy. There was actually a professor from the Ohio State University came and talked about tissue engineering and 3D printing with stem cells. This is what I was thinking of. I got the 2 weeks mixed up. But he was talking about how there is a gentleman, I think in Sweden, maybe in the United States, but I know there's a company in Sweden that does this. But they take these cells, drive them back, and their goal is to make these things called organoids. And it's not necessarily for implantation. You can use them for implantation, but there's a lot of hoops to jump through in order to get into humans. But what they do is they grow from like a very few stem cells, and they drive them in a direction to create like a piece of an organ. So like a piece of a liver, a piece of a lung, all this kind of stuff. And they use it for drug testing.
Wow.
They use it to test all these different drugs that are specific for those tissues to see if they're actually going to work, how toxic they're going to be, how much they're going to harm the cells, if they even have any, effect on the tissue at all. And I think that is so cool that when we talk about stem cells, it's not just all for humans, right? It's for more testing and more data and more results and like better results, because you can only get, mice are not men and men are not mice. Okay, that's something that I heard in a lecture a couple weeks ago, and I really like that because we do so much animal modeling, and like mice organs are close, and pig organs are close, and dog organs are close, but like you're never gonna get to test on a human until your phase 1 trials before you've even proven your mouse models. But your drug could work amazing on a mouse model and then do terrible in humans because we're not the same. We don't have the same number of chromosomes, right? So these organoids and using these iPSCs to create neurons and create muscles and create all these different cell types are not only going to change how we look at human medicine by like implantation, but also how we test human medicine.
Yeah.
And how, modeling with mice might be thrown out of the window in 10, 15 years because there are going to be these banks that instead of selling mice, they sell organoids for you to test your drugs on.
Now imagine if you can also do this for allergy testing. You don't prick people.
Yeah.
You just
throw
take a
it on
sample
yourselves.
of their skin, put it on a chip or something, and then
Yeah.
throw some medication on that.
Yeah, you'd have to make sure you get the immune system in there, but that would be really cool.
Yeah.
But I think that the potential for iPSCs and mesenchymal stem cells as a whole is really interesting, and that's why we do this. We want to share, and we're not going to be right every time, but we have ideas, we have fun. It's fun. It's supposed to be fun. Science is supposed to challenge you, but it's also supposed to bring enjoyment.
Exactly.
So, all right. Well, got anything else for today?
No, I think that's all for
All right,
iPSCs.
perfect. Yeah. Well, I just want to say thank you guys for watching. My name is Nick. That's Heem, and this has been Cells to Cell. See you guys.
See ya.